Greetings, Dr. Jinkai. I am Naufal, and my group consists of Krishna, Qingliang, Sylvester, and Kurt. We are Group 1 from Class O2, and today we are explaining all about proteins. So you might be wondering, what are proteins? Basically, they are essential nutrients of the human body as they provide energy required for activities such as running, working, and playing music. They are also made up of amino acids, which is represented by these circles. So, where do we get the amino acids? Well, we get them in our diet from things we eat, such as eggs, salmon, and chicken. Once we eat this food, it will pass through our digestive system, which is represented by the picture on the right. Once the food we eat containing protein reaches our stomach, the proteins will be broken down into polypeptides. It does not stop here. The food will pass into the small intestine for further digestion, where polypeptides will be further broken down into amino acids, and this is how amino acids are obtained. There are a total of 20 amino acids, but today we are looking at 5 basic ones, which are lysine, alanine, glutamine, aspartic acid, and threonine. Now let's put these amino acids next to each other. This may look complicated, but let's find something common about these amino acids. On the left side, there is a nitrogen attached to 2 hydrogen atoms, and we call this an amino group. On the right side, there is a carboxyl group. In the middle, there is a carbon attached to a hydrogen atom, and we call this carbon alpha carbon. The only thing that is different is what trails off the bottom, and we call this the R group, which stands for the residue group. And this is responsible for the distinctive chemical properties of each amino acid. We build protein in our body using amino acids, and we use something called dehydration synthesis to do that. So when you place one amino acid beside another, you can see that there is water. This water is lost during this process, forming a covalent bond between 2 amino acids. This bond is also known as a peptide bond. And what we have here is a polypeptide. Each of these individually are called a peptide, but if we attach them together, we get a polypeptide. Different R groups gives different chemical properties to each amino acid. Threonine is going to be polar, meaning that it is going to have a charge. Alanine, on the other hand, is going to be non-polar, meaning that it does not have a charge. Why is that important? Well, if being polar means that it is hydrophilic, this means that water is going to be attracted to you. Thus, you find threonine in the presence of water. What you end up getting is a 3-dimensional protein. The middle part, which is the brownish tan part, is the backbone. This is going to be made up of all the parts of the amino acid that are the same. So the alpha, carboxyl group, and amino group will be repeated over and over again. However, all these things on the outside that are trailing off are going to be the R groups, which are known as residue groups. And these are the groups that are going to fold off the end. This will lead to protein folding. This right here will be a polypeptide, which is a number of amino acids attached together. Usually, there will be thousands of amino acids in a typical protein such as hemoglobin, which is found in our red blood cells. So what will happen next? This will fold into a 3-dimensional shape. Polypeptide is just going to be this sequence of those amino acids, and once it's folded into a specific shape, then we can call it a protein. Next, we will talk about the levels of structure in a protein. To give you a brief summary, there are 4 levels of structure in a protein. Firstly, the primary structure is going to be the order these amino acids are bonded together. The next thing we have is what is called alpha helixes and beta pleated sheets, and we call that the secondary structure. What it looks like is represented by the pictures on the right hand side. So, the beta pleated sheets are going to be 2 sides that are attached to each other, and the little dots you see in the middle is going to be the hydrogen bonding between adjacent sides of the polypeptide. Then, we have the tertiary structure, which is the 3rd level of the structure. It is going to be those R groups interacting, and so maybe we have one that is hydrophobic. There are disulfide bonds and the positive attracting to the negative. Lastly, we have this quaternary structure, which is when we have one polypeptide attached to another, or a protein attaching to another protein. Hemoglobin is one example of that, which is made up of a different number of different subunits. Now let us take an in-depth look at the primary structure of the protein. Primary structure refers to the sequence of amino acids in a polypeptide chain, and it is the most important of the 4 structural levels, as it determines its overall shape, function, and properties. When the carboxyl group of one amino acid reacts with the amino group of the second amino acid, a peptide bond and an amide linkage is formed with the elimination of a water molecule. Now, this is a dipeptide bond formation, a formation between two peptides, and this is how the different terminuses are identified. Important things to note: primary structure reflects the relative positions of the residues. Thus, peptides containing the same amino acid residues but in different order are not identical. Proline-lysine is not identical to lysine-proline. Let's move on to the secondary structure. This refers to the way in which segments of the polypeptide backbone orientate, forming a regular pattern through the bonding between the groups. And these have two patterns, alpha helixes and beta pleated sheets. So, what are alpha helixes? Basically, they are the coil arrangement of the primary structure, and the coil is stabilized by the formation of hydrogen bonds between C double bond O group of one amino acid residue and the NH group of the amino acid residue 4 residues away. For further understanding, there is a picture on the right hand side which helps you to visualize the arrangement of the alpha helixes. Beta pleated sheets, on the other hand, looks like this. They are formed when segments of the protein structure chain fold back on themselves, and they are basically polypeptide chains aligned side by side in rows which are connected by hydrogen bonding between all the peptide linkages. The R groups are projecting above or below the sheet perpendicularly. There are two types of beta pleated sheet structures: parallel as well as anti-parallel. Parallel beta pleated sheets refer to adjacent strands which run in the same direction, while anti-parallel refers to adjacent strands which run in the opposite direction. Anti-parallel sheets are more stable as compared to parallel sheets. Let's move on with the tertiary structure. This refers to the alpha helix or beta pleated sheet folded into a specific 3D shape, and this fold is stabilized by various forces. There are two groups of proteins, fibrous and globular protein. Finally, we have reached the quaternary structure. This refers to the special arrangement and association of more than one polypeptide chains or subunits. It is also referred to as a combination of several protein chains into a larger 3D structure held together by R group interactions. So you might be thinking, what are the uses of proteins? Well, proteins act as enzymes. Enzymes speed up chemical reactions in the body and play a role in many activities happening in the body. Hormones are also proteins, and they control blood sugar concentration and activate our muscle growth. Lastly, proteins act as antibodies which protect the body against viruses and diseases. To end off on a high note, here's a fun fact. Did you know that human cells contain about 1 to 3 billion proteins? We have come to the end of the lesson. We hope that you have enjoyed and gained a little bit of knowledge from this short video. This is Group 1 and we hope to see you again. Thank you.